My name is Cotsapas. This is my lab looking very smug because we managed to beat the Escape the Room, the hardest room in Escape New Haven, back in the before time when we were allowed to be together. And we work on a diversity of problems, and I kind of put together sort of what we do and who we are. So we really try and approach big questions, mostly in disease biology. We use a variety of techniques and technologies. We do whatever it takes, pretty much. We use and we develop both robust and quantitative methods to look at things. We're very collaborative. The things we primarily value rather than expertise is curiosity, drive, and inclusivity, because expertise is something that gets built by domain experience, we've found. And we have a really positive environment. We're honest with each other, sometimes very direct, but we're also very kind with each other. And I think Jason's question actually sets up a major theme in the lab. So I'll come back to that in a second. I also asked the lab what they thought we did and how we should describe ourselves. And I got a diversity of opinions on Slack. A bunch of grumpy nerds, a dynamic intellectual environment where input is valued from all. So I came up with a dynamic, forward-looking group where grumpy nerds are always welcome to give input, but sometimes don't because we're introverts. I also want to point out that we have a dedicated D&D channel on our Slack. Draw your own conclusions. So we work a lot on autoimmune and inflammatory diseases. These are a group of almost 80 diseases where basically the immune system decides it doesn't like some part of the body, which it normally tolerates. If it doesn't like the myelin sheath around your brain, you get MS and you get autoimmune attacks on the brain. You can have attacks on skin components, you can have attacks on gastrointestinal and other internal organs, on salivary glands and tongue, on the pancreas, on the skeletal system. Basically, there is a diversity of lack of tolerance. Like most complex diseases, these are highly heritable. We've been doing GIWAS in this space for about 15 years now, and we've gone from the first association 15 years ago when I was a postdoc in rheumatoid arthritis to now having almost 1,000 loci across maybe 10 diseases. These are very large efforts across large international consortia. We play in that pool quite dramatically. The latest couple of papers have been in MS up here, where we've got a couple of hundred loci and a bunch of rare variants. And as Joel also mentioned, the big game now is, what does this stuff mean? And where do we go? How do we infer biology from this? And so the big realization, almost 10 years ago, we wrote this paper with Matt Morano and John Stamm, basically showing that GWAS hits were enriched in regulatory regions, in accessible chromatin that looks like gene enhancers and gene promoters, not in the coding regions. And that has really stood the test of time across all traits. We're particularly interested in the autoimmune diseases, but this is true everything from human height to autoimmune disease, to metabolic disease, to psychiatric disease. It seems to be how complex disease works, excuse me. And so this sets up a problem, sort of how do you infer what's happening and what the biology is, what cell is being perturbed, what the function is happening. And so that's a question we've been asking ourselves. And so the model you might imagine is you've got some regulatory element that has a SNP in it, and this regulatory element controls some gene. And so if you have two different alleles in that regulatory element, and that's the causal allele, you might expect that you get differential regulation. The most obvious one might be changes in expression. So when we try and find map, which is difficult across diseases, we can sometimes get to a single element. We've built techniques where we can find map all of the GWAS data down onto regions of accessible chromatin, we can prioritize them with posterior probabilities and say, we think the signal is coming from this element, and we think this element controls this gene. And so we've built methods to do this. And we can usually find map about 30 or 40% of loci in this way. The other way you can do it is you can say, well, if the risk variant is perturbing a gene, then the gene expression must be a genetic trait. It must be an expression QTL, and those are very common. And they must look the same. It can't just be an EQTL that's nearby, they have to look the same. So whatever's driving disease is also driving expression. And if you look carefully, you can see very clear cases where you have a GWAS peak, in this case for MS risk, at the top in black. And then when you look at the association pattern in the same region for a different trait, in this case for the expression levels of a gene called metal21B in T cells in the middle and in monocytes in the bottom row, you can basically see that these three signals are identical. You don't have to squint that hard to see that. And that's kind of intuitive, but it turns out that it's harder than that. Because if you look up at the top, those are all the genes that are in that region. The ones with gray bars, we just can't detect in any of the cells we look at. The ones with black bars all have EQTLs in that region. There is an association between a genetic variant and the expression of each of those genes. And so if I was just picking which gene would be interesting in the region, all of them would look convincing. But almost none of them look like this, where there is a consistency between the disease signal and the gene expression signal. And so you have to work a little bit harder to infer this. And what's surprising here is, that we can only do this for about 25% of loci. And in the other 75% of loci that we look at across hundreds of loci now, we can't find evidence for a gene expression trait that matches the disease. And so there's a big question in the field right now, what's going on? We believe the observation that most of the heritability of these traits is landing in regulatory regions, but we can't really look at individual loci and find a gene regulatory event or a gene regulatory region that drives the signal. So there's a disconnect here. And that's a big question in human genetics. And so we're trying to solve that. We have several ideas about what to do about that. Part of it is, I think we're looking at the wrong cells under the wrong stimuli, under the wrong conditions. And so we have to look at those. And part of that is we have to take multiple different kinds of molecular measurements and build a much more complex regulatory model for each gene. And so we're pursuing that. And doing that just on paper seems simple. Building robust statistical methods to do that is much harder. So the other major problem that we're working on is, I showed you this slide earlier, and there's a reason that I put a Vitruvian woman in the center of this rather than a Vitruvian man. And that's because autoimmune disease is very sex biased. Most of these diseases occur much more frequently in women than they do in men. A handful of them occur much more frequently in men than they do in women. Some are not biased, but overall, 80% of autoimmune disease diagnoses are in women. And we have no idea why. People have tried, people have looked, but we have no idea why. So we're starting to ask, okay, what are the differences between men and women? That sounds both stupid and obvious, but when you start thinking about it, there are so many differences between men and women that isolating one that is relevant to disease and maybe relevant to the immune system is actually quite challenging. We are different in terms of our capacity to bear children and the effects of pregnancy on our bodies. We are different in terms of the amount of various steroid hormones that are sloshing around our systems and affecting pretty much every cell in the body. We're different in the terms of the number of chromosomes and the type of chromosomes that different people carry. And we may be different in terms of the effect of autosomal variation on traits. We know, for instance, that there are different effects in adiposity and body mass index. Some risk variants only affect males and not females, and vice versa. And we don't know why. So we're working on this. That's a sort of fairly major stream in the lab, and we're taking a diversity of approaches. But one of the things I just wanted you to leave you with, because we also do experimental work to test our hypotheses, is this observation. Basically, wherever we look, we see differences between men and women that I think were not known before. And I think they were not known before because people didn't look. So what I'm showing you here is what happens in T cells in terms of signaling when you hit the T cell with a stimulus. In this case, the stimulus is TNF. That's a really standard way to hit T cells, which are a component of the immune system. It's not particularly important what they are. But basically what happens is you hit the cell, you get the ligand, the stimulus binds to a receptor, that signal gets transduced inside the cell, you get some signaling event, and then eventually that goes to the nucleus and you get a bunch of gene expression consequences. This is about how much signaling happens in response to the same dose. So the dotted line is what happens at baseline. I just hit the cells with TNF and I get an increase in signaling, which is exactly what you expect. The solid line is what happens if I've already exposed the cells to estradiol. Estradiol is the most common form of estrogen that's present in the human system apart from pregnancy. So under normal conditions, both men and women have estradiol. Women have much higher levels of estradiol and it varies over the menstrual cycle, but men also have some estradiol sloshing around. But what I want you to see is that the baseline signaling is different when you expose cells to estradiol, and the slope of the response is different. So estradiol seems to blunt the ability of T cells to respond to stimulus. What the consequences of that are, we're not sure. But it appears that if you are a T cell that lives in a high estradiol environment, such as a female system, you may be responding differently to stimulus than if you are in a low estradiol system. These are pretty fundamental differences in the immune system. What the consequences are and how that plays out in terms of disease, we are trying to figure out. And I will leave you with that. I know it's hard to ask questions